Okay, we're at 10 o'clock. So we would like to welcome everyone. Thank you all for joining our discussion on clinical trial readiness at Boston Children's Hospital. For those I've not yet met, I'm Ann Lenzen, the founder of the US MEF2C Foundation. We have some team members here joining us as panelists to help sift through questions, and I'll let them introduce themselves. Do you want to start, Tushar?
Hello, everyone. I'm Tushar. I live in New Jersey. My daughter Myra is 7. And I think it's great we could meet everyone today. And I think we are taking the first step in the right direction. And hope we continue this journey together. And we like all the support we can get from all of you guys. Thank you.
Caroline?
Hey everyone, I'm Caroline Claflin. I'm on the Parent Advisory Board for the USMEF2C Foundation. My son Darwin is 5 and I live in Boston, so I'm really close to Children's, which is great. But I'm really, really happy you could all join us this morning and really excited to hear more from Dr. Chung.
Stephanie?
Good morning, everyone. I'm on the West Coast, so my camera's off. It's a little early here. I'm on the Parent Advisory Board also. My daughter Demi is 9, and we actually live in Nevada right now, but we've lived in Colorado, Mississippi, and in England since her diagnosis.
Meredith?
And I'm Meredith San Antonio. My daughter Valentina is 2 years old and we live in Dallas, Texas. So I just wanted to thank everyone for being here. This exciting project is very important to the MEF2C community. Prior to our participation in Simon Searchlight, there was no official patient registry which the scientific community could access. In addition, of the research assessments will help us compile more data on our patients, which is critical in creating a better understanding of the community. This project will provide parents the opportunity for their child's specific variant to be recognized and bring us together as a community. So we would like to welcome Dr. Wendy Chung, a leader in the rare disease arena, and joining Dr. Chung is a member of her team, Alexa Geitzler, a genetic counselor at the Boston Children's Hospital, who will moderate the discussion.
Thanks, Meredith. I'm really pleased to be here, and this is largely an interaction, a discussion, as Meredith mentioned, and so we'll be wanting to hear your questions. I'll start out by describing a few things, but it's not a formal presentation. As I said, we hope this is a discussion. So the first thing I'll say is that for those of you who don't know me, I'm a medical geneticist, a pediatrician. I was at Columbia for actually about 25 years, but have now been at Boston Children's Hospital as the chair of pediatrics for about a year. In making this move, it was quite intentional to be able to think about what the community of rare diseases needed. And I'm very proud to say that many of the resources that are at Boston Children's and many of the resources that are around Boston generally, this is across Harvard Medical School as well as in the biotech sector, are really coming together in a strong and powerful way to think about treatments and moving forward to improve lives. Within this, I find that it sometimes requires some being able to bring people together and to realize what we can do to swim in the same direction and to appreciate many of the opportunities. And so that's what I'm trying to do, is act as that, in some ways, catalyst, in some ways, convener, in some ways, being able to partner you all with the other scientists, the other doctors can help us to get to where we need to go. We're not there yet, but we need to prepare for that. And the reason I say that is I'm watching some of my other groups that I work with in different phases, and I've learned from them and continue to learn from them. And I wanted to make sure you all could benefit from that experience. And even though you don't do things right always the first time, we hope that we learn from those experiences and that you suffer less of the growing pains that I feel like we've gone through with some of the other conditions. So sometimes being first out has advantages, challenges. And I'm very pleased to say that as an example, we had another rare disease community that came to meet together in Boston last weekend, and we were announcing our first clinical trial for that community that'll be starting by the end of the year. And so it's by taking some of those experiences, as I said, and building upon them that I think we've got many opportunities. I won't go into a lot of detail, but I'll say that Anne and others have been planting the seeds in the basic science of what's needed to be able to move forward to clinical trials into therapies. And even as recently as last night was meeting with many of the neuroscientists at Harvard to be able to think about strategically what we need to go forward in terms of being able to de-risk this and to plant that foundational work that needs to go forward. And so in doing this, I'm trying to represent a lot of the pieces of what's going on, and I'll be glad to answer the questions in that way. What we all need to do as a community in terms of taking care of each other are a couple things. So I think about one of the slogans I sometimes say is the care until the cure. I want to be careful in terms of using that word cure too audaciously. So I want to set expectations and realize that I do think there are multiple ways that we can improve lives and improve care. But one of the things is learning from each other. So just simply being able to understand what someone has done that may have worked well. And if we see that in more than one case, we start to see that pattern and start to think about how we can apply those learnings from one person to another person. Within that, it involves collecting information from as many people as we can and understanding and evolving what I think of as best practices or clinical care guidelines, things that we can have evidence to be able to say, yes, we see a pattern. We think this pattern actually is something that we can use across individuals, across the United States, across the rest of the world. And in doing so, sometimes you all need ammunition to be able to get people to pay for it or to be able to get services in schools or educational settings. And so having these practice care guidelines or clinical care guidelines is a way, I think of it as kind of an owner's manual that helps your pediatrician, your neurologist, your developmental pediatrician, your educator be able to see what are the things that they may be able to do to support you and your child and to be able to hopefully allow them to be healthier and achieve more. So one of the most concrete near-term things that come out of the things we're going to talk about today is clinical care guidelines and standards. And with doing this, hopefully updates in those over time as we develop improvements that we can make. Another thing is what I would call clinical trial readiness, and that's a lot of what we're talking about today. And I can say that this is one of my learnings based on interactions with the FDA, interactions with other researchers, and thinking about things. And that's that our children, in a good way, are developing. They're not static. They're gaining skills over time. They may be gaining skills more slowly than other children. So if other children have a slope that's going up like this, their slope may be less than that in terms of their gaining skills. But when we're looking at an intervention, be it a medicine, be it a genetic therapy, be it a new learning device, whatever it might be, what people are looking at is, are we changing the slope of that curve? So are we going from here to here and improving things? Because they always ask me, well, maybe that change that you're seeing would have happened on its own anyway. How can you be sure that it's actually the intervention that you're doing that's responsible for that? And in part, the answer to that is we have data to show us this. We have enough data points going along and over time for your children that we know what their trajectory is like. And it's not just your child in isolation, but it's many other children that we can be quite confident that the slope is this and not this and not this. And so within that, we think about that as that clinical trial readiness. We also think about, as we think about outcome measures or what would change with an intervention, we need to know what's important to you. What would make a meaningful difference in your daily lives? What would be important? What would make just literally getting through the day easier? And we also need to inform people at the FDA about what those things are so that they get their heads wrapped around it and start to accept that as being something meaningfully different. And so as an example, it might not be that, this has to be completely cured with a capital C in terms of taking someone from their current level to quote unquote neurotypical. critical, but things that might be small improvements in sleep, in daily function with communication, in things towards independence, in outburst or behavioral sort of difficult, challenging situations. Any one of those, you might say, would be something that would be a significant improvement. And so we need to understand from your perspective, what are those things that you would find important to improve? And then how do we measure it? And that becomes a key, is how do we actually collect data in a way that you can tolerate? We don't want to torture you. We don't want to torture your child. But how do we collect data? How do we understand that the FDA will accept the data that we're going to collect because they say, no, this is something that we can rely on. We know that it's valid. We know that it's accurate in terms of the measurement. And with this, we may have to have other, I'll call them surrogates, but other ways that help us to collect the data. So there may be some things that we need to do in person with an evaluator that's objective, that's trained, that's reliable to do this. And that evaluator, we may also want to complement with information that you're reporting from home, because you may see different things at home in a naturalistic setting than what we see when you come to Boston Children's Hospital or some other hospital or clinical research setting, where, to be honest, your child may not be their usual self. They may be a little off their game. They may not sort of behave because it's unfamiliar. It may feel different. But if we can see the alignment between what we see with objective measures, people that are trained to do this, and that correlates well with your reports of things from home, we may be able to gather enough data to present to the FDA to say, look, we have anchored this here, and we can now take a parent or guardian reported measure, and that may be as good or even better than what the FDA has previously accepted as an outcome measure. And so in doing this, though, we need to gather that evidence, we need to practice, we need to do some presenting of this information back to them so that they can be they can be with us in these planning stages for those trials. One of the things, though, that can sometimes happen is, in a good way, things unexpectedly can fall in your lap. And I have to be honest, I don't know when some of those things might fall in our lap. And so it's always better to be prepared. And in doing so, it takes time to do this. It takes time to organize ourselves as a community. It takes time to work out the kinks. It takes time to train people in the right measures to do this. So with all of these things, we're trying to be sort of ready. We're trying to be kind of the Boy Scouts and always be prepared for these things, so that if something should fall in our lap, and as an example, if one of the medications that we're using for another condition should be successful and should work, we might say, well, we'd like to see whether or not this would work for MEF2C. And if it would, then we'd want to be able to be prepared and have those baseline data, those outcome measures that we could test them right away. There may be things that might take more time, that might be very, very specific to MEF2C based on some of the fantastic research that the neuroscientists are doing on MEF2C. And again, if they should have a breakthrough, we want to be at the ready to go in terms of anything they might come up with. And if there's even something like breakthroughs in genetically based therapy or gene editing, we want to be able to, again, be right there. So I know this is a little bit amorphous. To be very clear, I'm not saying January 1, 2025, we're starting a clinical trial. Just to be clear, I am not saying that. We're not in that position yet. But for many other conditions that I've worked with, it's taken us 2 to 3 years to even be able to get the first people ready for that clinical trial readiness. And so we're starting that process now so that we can be ready if any of those things should fall in our lap, either because of the intentional work that people are doing or just by chance something is proven to be effective. Within this, and I say this because there could be opportunities, and as our children are getting older, I don't want to be a Debbie Downer, but in general, my thinking that I think is probably true is the sooner we can get in, the better. So a younger a child is, and the sooner we can get in, the more opportunity we have to help them be able to change the slope from this to something more like this. And so I just don't want to lose what could be precious years for your child by realizing after afterwards that we have to kind of get our act together. And I don't know how much time we have in terms of windows of treatability, but I want to make sure that we don't leave anyone behind in this process, and that as we do this, we can be nimble, we can be agile, and we can be quick. The other thing I'll say is that as I look around other people and what they're thinking about working on, they are sometimes doing a survey to see what conditions are out there, what communities are very well organized, what the frequency of the conditions are, and who's really shovel ready is the term I'll use. In other words, who's ready to go out the gate. And if we can be at the ready and show that we're good partners, that we could execute quickly, that we know what we're doing, I am seeing different people say, well, I could do X, Y, or Z. If X is farther ahead and if they look like they've got a parent group that's well organized, we're going to go with X because we know we'll be able to get that done first. And so I want to have you all positioned in a place where you're at the head of the line in terms of being able to go forward. And it just, I think, will be show a demonstration of good partnership. So what does that partnership look like? Let me be clear, and I know it can be a little bit confusing in terms of what's the exact ask here, Wendy? Like, tell us what to do, we'll be glad to do it, but just what are you talking about? So there are two components to this, and maybe three components, depending on what you exactly want to do. One is that there is an online study called Simon's Searchlight, and this is not exactly the natural history study, so let me try and break it down. So Simon's Searchlight, I'm the what they call principal investigator for, and I started that study 14 years ago, so quite a long time ago. It's been going strong, and it is international. And so people around the world in many different languages can be part of Simon's Searchlight. Alexa was one of the genetic counselors on Simon's Searchlight for many years. And again, that is an online study. And so the burden in doing that is much less in that you don't have to travel to Boston Children's. You can do that from your living room at 10 o'clock at night after the kid's in bed. You can do it in pieces, little bit by little bit. And with that, you have the opportunity, if you'd like to talk with a genetic counselor and ask some questions about MEF2C, but that is a longitudinal study, in other words, happening over time. So we're watching how individuals change and develop over time. That to me is the foundation. Everyone ought to be doing that. And if there's a reason you're not doing that, we'd like to hear how we can make it easier for you, better for you in doing that. As part of the value of doing that, what we do in Simon's Searchlight is we have reports that go out to everyone in the community so that we're learning together. You can see what we're learning. You can see, I hope, what are easy to understand infographics about what we're learning so that you can take that information to your doctor. You can share it with your partner, your in-laws, your parents, whoever you might want to, but that you're seeing all of that very transparently and you're seeing it together. And that information is de-identified, but then shared with qualified researchers around the world. And so that actually leverages your input to give to many, many people, but you only have to do things once. You don't have to do them over and over and over again. You can enter once and be used very widely. All of that information is great, but it suffers the limitation that it is reported by you. So I'm going to get back to what I said before. There are some people, I am not one of them, but there's some people that say, those parents, I don't know if I can trust them. What they're seeing may not be what's actually true. And so we need to have independent confirmation of what they're seeing, that they're actually reporting correctly. So the second part of this is what we've designed in terms of the in-person assessment. So that's an anchor for a subset of individuals who I hope are already part of Simon's Searchlight to then come and have a direct assessment where a qualified researcher who's reliable, who's been trained, who's been certified, who the FDA believes in these measures, does some of these measures that the FDA and the EMA in Europe know are things that they think should be part of clinical trials, and we can directly assess those and over time be able to see if those are changing so we get the slope of that curve. Those direct assessments we think are things that are going to be easy to be able to incorporate into a clinical trial in a way that the regulatory agencies like the FDA, the EMA will accept. So those we feel like are a slam dunk for the FDA and the EMA, but I have to tell you that I'm worried that those may not be our best measures. Again, it's what the regulatory agencies have traditionally used, but I worry and wonder that you actually, because you're with your children every single day, because you know your children best, because your children are most comfortable at home, I wonder whether you might actually be better assessors in terms of what's changing. So the combination of things that you're doing plus the combination of things that we're doing, I think are the secret sauce in terms of putting those together. So a subset of you who are willing to actually go into a clinical center, and I know it's a sacrifice to do this, but to be patient with your child as I hope we don't torture them, but put them through some of these, I hope they feel like games, but they are tasks in terms of being able to see how well they can do. Doing things like putting on the cap for an electroencephalogram, so things where we can directly measure their brain waves, and objective measures like that that you can't fake. So something like the EEG is something that is not sort of reliant on the observer. It doesn't matter whether you're doing, it doesn't matter who the observer is. It's direct brain wave data that's coming from the brain. And the FDA likes things like that that are objective. So we're going to be doing things like direct EEG collection, looking at how your child moves. So what we consider motoric, how they walk, how they move their body, how coordinated they are in doing that, how they use their hands in fine motor skills, all those things that are important in terms of daily function, and being able to assess that in a direct, objective way by using image analysis and by doing things that we can directly see those movements and coordination and quantify them over time, or other measures. Eventually, I'm not saying that we're going to do it right away. We are also exploring with some of our other groups other things that might be important to you, and this can be part of today's discussion. What are those other factors? So I'll give you an example. If sleep is something that you think is challenging and being able to look at how we can improve sleep, we're developing ways of being able to assess daily or nightly, I should say, sleep from home to see whether or not you get a child is getting a good night's sleep. Whether or not it's interrupted. And we've chosen sleep as one of the things to think about, because if you get a good night's sleep, it tends to be that you learn better the next day. You have less behavioral challenges the next day. It's the time when a lot of learning and memories are consolidated. And so sleep is kind of a foundational thing in terms of the brain and behavior. So we prioritize being able to assess sleep. You'll tell me whether or not you think sleep is important, but for many conditions, many neurogenetic conditions, we think sleep is important. There may be other things where we can have you take videos from home and send videos back and upload them, that may be a way of being able to get more data points without having you travel to a clinical site. So if you're a long way away and it's a big deal to be able to travel, what can we do where you can be our eyes and ears in collecting the data, and we can analyze it centrally, but you can collect it remotely? So we're exploring those as what I would call the third leg of the stool, to think about the multimodal ways, or the various different ways of collecting those natural history data. So that's what we're trying to prepare for, along and in parallel with what many of our basic science teams are doing in terms of developing those treatments, be they pills, be they things to manipulate genes, whether it's gene expression or the way the gene functions, or even direct editing for doing this, but in parallel with what they're doing, so that we can meet up at the same time, so that as soon as anyone's ready with one of those therapies, we're ready to take off at the same time that they are. I'm going to add one further complexity, which is that for many of you, if we were to put up on the screen all the different mutations that your child has in MEF2C, there's quite a range. And within the range, it is possible that there's going to be some differences depending on the underlying genetic variant. And so I personally think it's important for people to stand up and be counted, stand up for themselves. We may find some clustering, that those guys over here can sort of cluster together, and that they'll be able to represent each other. And there may be other clusters over here and other clusters over here, but I don't know for sure. And it's possible that the baseline data that you're entering and collecting now will be for your own child over time, their own baseline. So by doing so, you're essentially preparing yourself for a position in a clinical trial because we know what your child's trajectory is going to be. Now, that's not to say that if your next door neighbor is diagnosed with MEF2C this Christmas, that they won't be allowed to be in a trial, right? I mean, they can enter whenever they're diagnosed and do that. But I do think the individuals who are sort of, standing up and being counted for themselves are going to be in the best position in the long term to benefit from the standards of care, from the clinical trial readiness, and ultimately for the clinical trials. So from a selfish point of view, I think it's in your own best interest to do something. And I think what we're trying to figure out collectively for the community is how do we work together, help ourselves, help each other as we're doing this, but have at least enough and broad enough representation across individuals across the age spectrum, by sex, by different genetic variants, to make sure that we have a good sort of broad assessment of what the community representation is, so that we're not leaving, any part of the spectrum out as we think about the future trials. So let me pause there, and I know some questions are coming in the chat, so I'll let our team just throw out some questions in whatever order they think is important.
Yeah, so starting with a clarifying question about something you had said, is that will we be clinical trial ready in 3 years, or will we be ready to start the process of clinical trial readiness in 3 years? And now we're working on kind of the pre-clinical trial readiness activities.
So I consider this phase the clinical trial readiness phase. I don't know that it's going to be, just to be clear, exactly 3 years. I'm not sure exactly when a clinical trial opportunity will come up, but I think we're doing this now. We're doing this and have been doing this, I'll say, for instance, with the Simon Searchlight Registry already. So we're not starting from ground zero. We've been populating that for quite a bit of time, some of you for years even in doing this. We're sort of, I would say, going into the next phase in terms of being able to do these in-person assessments, which are the second leg of the stool. And over time, I hope we'll be able to add even some of those remote measures, because I do think I don't want to leave people behind. And I know that in-person is challenging, just in terms of time and commitment for families, depending on where they are. So as we're doing this, we'll continue this and expand this and learn from other groups as well who are doing some similar things, even for different conditions, but that share some of the same challenges as MEF2C.
Okay, and another question we have mentioned that the consent forms for the ASCEND study mentioned that this research is being funded by other organizations that work on other genes. And we want some background on why are these groups interested in MEF2C, and are these genes related to MEF2C?
Yeah. So I know this is a bit complicated, but in my laboratory at Boston Children's Hospital, our ASCEND protocol covers a variety of neurogenetic conditions. And in part, this is administratively just easier to do. So we have one umbrella protocol that has the same assessment. So people who are getting EEGs, people who are doing a Vineland, people who are doing these walking studies that we do and fine motor skills. So because all of those are the same, we've lumped them together. And so there's MEF2C, there might be another condition, and you can go on our lab website to see some of the other conditions we study, but they're all neurodevelopmental conditions that are caused by a gene. Each of those sort of group condition things might be funded by that group or by a grant that funds those group, and we keep the funding separate for each group. So each, if someone were to donate to MEF2C, the funds only go to MEF2C in terms of doing things, but there is an economy of scale. So as an example, if I have a coordinator like Alexa or Jessica Waxer or other people, I don't have to have one coordinator for each condition. I can use a coordinator who's brilliant and understands how to organize these studies and be able to use a portion of their time for MEF2C. And I don't have to spend as much money for a larger staff having individual people devoted to each study. So that may sound a little confusing in terms of things, but at the end of the day, it's a more efficient and effective way. And it also helps to just smooth things out so that certain groups, for instance, may have a great time in fundraising. Like 1 year is a great banner fundraising year for them, and so they've got funds. I wouldn't want to have to hire and fire a whole study team just because it wasn't a great fundraising year for one group. I want to be able to smooth things out and keep the momentum going for conditions. So that helps in terms of just longevity and evenness in terms of what we do.
And we have a question about treatment mechanism. Basically, can you make the mutated gene turn on, and or can you have the gene copy that is working to produce the additional protein needed?
Yeah. So I'm not going to go too much into this, but to say that for some individuals, we think the mechanism for MEF2C, this is a complicated word, but is due to haploinsufficiency. So what I mean by that is that all of us have 2 copies of MEF2C. For some individuals, the thinking is that the genetic variant causes 1 copy not to work. And so instead of 2, it's haplo or half as much, or insufficiency, meaning not enough and not working. So for a whole bunch of that MEF2C community, the thinking is that the mechanism is all the same, that there's still 1 good copy working, but that the body is detecting that it's not enough. And so the body would work better if, in fact, we could either fix the copy that's not working, or the other way that people think of it is make this copy, this good copy, work overtime. And in working overtime, it would compensate for the other copy of the gene that's not working. So people have thought about this because MEF2C is not the only brain gene that works this way, where we think haploinsufficiency is the problem. So that's what I was saying in terms of some learning from other conditions, is that another condition may figure out a way to turn up the volume for the 1 copy of the gene that's working better, make it work harder, make it work overtime. And if we can turn up the volume or make this work better, it may compensate either partially or completely for the gene that's not working. So people have thought about that either by using a pill. I think the pills sometimes are a little bit more challenging because they tend not to be specific to only affect MEF2C. They might affect other genes and turn up the volume for other genes as well. People have thought about using what we call allele-specific oligonucleotides, or ASOs, to be able to pump up the volume. I'm not sure whether or not that's going to work for MEF2C, but people have thought about that. And then people have thought about gene addition, being able to add back a copy of MEF2C to compensate for the copy of the gene that's not working. So there are multiple strategies, I use the term shots on goal that people are talking about. I think for the purposes of what we're thinking about today, in some ways it doesn't matter which of those might come forward. We want to be ready for any and all of them. That's what I mean by clinical trial readiness. There's a second point, which is that there is thinking that maybe not all MEF2C mutations work in the same way, that maybe everyone is not haploinsufficient, that maybe in some cases the MEF2C copy that's not working might be doing something different. Maybe it's working overtime, maybe it's doing something differently. And in that case, if that is the case, that's a big if, but if that is the case, we might actually need to do something to fix this copy of the gene that's not quite right, or we might even need to turn down the volume in some way. And so there are a lot of people working on the basic molecular mechanism to understand in all cases, how this other copy of the gene is working, what it's doing, what we need to change for doing that. So in parallel to what we're doing, we want to be able to have them do that basic science. When it comes to the clinical trial readiness, I would argue it doesn't matter. It doesn't matter whether we know the molecular mechanism for your child's mutation. We're going to have to do the same thing for clinical trial readiness regardless. And so collectively, it doesn't matter what MEF2C genetic variant your child has, as long as this is the genetic answer for why your child's development is different. We need to come together to work on this, because I think the MEF2C researchers are looking at us together. They're not looking at us as separate. They're coming for one-stop shopping, if you will.
And I think that answers another question that someone asked. Is this study kind of a broad MEF2C gene study, or a MEF2C haploinsufficiency syndrome study? Are they the same or different? Does it matter as we look to, different variants?
Yep. So it is broad and inclusive.
And we also have a question about if any medications are being used or tried right now. This person has a question that their daughter has had a skin biopsy that's growing with Dr. Lipton with the goal of introducing agents and wondering if any medications are being used.
So I can't speak for Dr. Lipton, but you can certainly reach out directly to Dr. Lipton in terms of what he's thinking. I do know he's working on things. There's nothing in clinical trials for MEF2C. There are things that I think individual families are using symptomatically with approved medications that their doctors are prescribing. But there's nothing in a clinical trial and nothing to my knowledge from Dr. Lipton's lab that's in a clinical trial today.
Let's see, and we have some kind of logistic questions like how many patients are we looking to attend clinic for this study?
So in doing this, I think we're looking for as many people as are willing to do this. As we go forward, we actually learn based on the spectrum of by age, by different ability, and so from a purely scientific point of view, we can make estimates that, for instance, it would be nice for 50 people to come through and to be able to know the spectrum of what we're seeing. And that generally is a good number in terms of understanding this, and 50 with the number. As we start bringing people through, we may make adjustments and realizing, you know what, everyone looks exactly the same, or you know what, there's a huge range in terms of variability. And so we may adjust those numbers. But if you want to put a, generally first pass target, 50 is a good number in terms of being able to know that we've got the, and again, by distribution across different ages too. We don't want 50 people who are all 3 years old. We want to have 50 people who span, hopefully, young children, adolescents, even if we can, adults.
And we've got a question about, is it still confirmed that siblings don't need genetic testing?
So for most of you, if you as parents were included in the testing and the mutation was found to be de novo, that is that neither of you as parents have it, and if the sibling, your other child, is neurotypical, they're not having any issues with developmental delay, then no, I don't think that child needs to be tested, because sort of if it's de novo, we think the two of you as parents don't have it to pass down to other children. There is a very, 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 I'll emphasize, very, very, very small chance that even with the parent's genetic testing and it appearing to be de novo, that it could be passed down in a sperm or egg cell that wasn't directly tested. So that what we call mosaicism in a parent is a theoretical possibility, but it doesn't happen very often. So again, if that sibling is neurotypical, they shouldn't need to be tested.
And we have a question that if a family is not able to make the October assessment, would there be a different date that they could come out or do it virtually?
Yeah. So for now, I'll say we're the October assessment, and just to be specific, so we've got a time that we're doing this in the near term to be able to do it in person at Boston Children's Hospital. I know we're going to learn a lot from that in terms of, just sort of right sizing the assessments and making sure we don't overwhelm the kids and that they can be their best as we're doing this. With that, we always iterate to improve these things. It won't be the last time we do in-person assessments by any means. As we understand both the value to families from coming, as we figure out what we need, as I was saying, and figure out whether 50 was the right number or whether we can do it with less, we'll have some better sense. And we have the facilities here to do this, really with enough advanced warning, at many, many different times during the year. So we can adjust to this based on what the community, what you guys need and what you want in terms of doing this. With this, in terms of thinking about this remotely, so Simon Searchlight as a remote way of doing this is already available today. That's based on standardized assessments online. Questionnaires is what I'm talking about, where you fill out questions and answer them online and submit those. The assessments I was talking about to do remotely from home that might be gathering videotapes or measuring sleep or things like that are probably not a 2024 activity. So they might be into the future, whether it be 2025 or 2026 is in part what you as a community feel are priorities, what I hear from you in terms of challenges, what we're ready and have off the shelf ready to go, and in part whether or not you guys want to be part of the early phase of testing out some of these devices and ways of doing things, or whether you want to wait until they're fully baked and others sort of do that within another group, another condition, and then you hop on board once they're more fully developed and fleshed out. So you'll let me know whether you'd like to be an early adopter or a later adopter, but we're certainly going to be doing this with my group with other conditions, but it doesn't have to be MEF2C if you guys aren't ready to do that or don't want to do that in sort of an early adopter phase.
And we had a question about how to join Ascend. And I think everyone can see the answers that I typed. I put in the Ascend study email that if you're not part of Ascend yet, you can send an email to that email address to get started.
And I'm going to say it one more time because I know I'm using a lot of different words. Ascend is the in-person clinical assessment. And in doing that, we try and do some homework ahead of time to make sure your MEF2C genetic variant is what we would say is associated with the neurodevelopmental issue, and understand things like the age of your child, the ability, so that we right size our assessment measures for your child. That is, that we don't come up with something that's too easy or too hard as we think about the assessments we're doing.
And let's see, would assessments be a day or would we need to stay over in Boston? And they also requested the link for Simon Searchlight, which I'll put in the Q&A as well.
Yeah. So in terms of overnight and things like that, my expectation is that depending on your child's, it would likely be 1 day. And the most I think it would be is 2 days. In doing this, I do hope that people can wake up refreshed and ready to, do the activities. So for some people, depending on where you live, that might be staying in a hotel the night before so that morning you can sort of come in, hopefully refreshed and ready to go. Depending on your travel and other things, that's why I said it could be 2 days, depending on exactly how your travel is. If your child were a child that 1 day would be too much, we have opportunities to spread this out over 2 days, like I said, so that it's not quite so overwhelming and that we can get an accurate sense of your child's ability. I wouldn't want to underestimate your child just because it was, too many new things all at once and it felt overwhelming. And I'm sensitive to sometimes some children may have those issues.
And does the study seek to involve patients outside of the US?
So in doing this, so number one, yes, we could take people who are in Boston for other reasons or came to the United States. We certainly would welcome them in terms of doing this. I actually, for this community, was recently in Barcelona and met with the MEF2C community in Spain, both the clinicians there and one of the family group leaders, and was talking with them both in terms of how to do something that might be in parallel so that families from Spain, for instance, wouldn't have to travel all the way to the United States, and that we could still be able to put the data together and have things that were being done essentially as a multi-center way of doing this. And I think that's an experiment. We haven't actually done it yet, but we've had some preparation in terms of thinking through doing this. And I think it should be feasible to do that. I think that would be a way of, again, knowing that this is a rare condition and we need to work together in terms of doing this. That model, I hope, is a model that we can replicate. Exactly how this evolves, how quickly it evolves, what sites we would do, very much a work in progress in terms of thinking through that. But we know your lives are complicated. Same is true for our lives too. And so we're trying to do this in a way where everyone can do their best and engage in a way they feel comfortable with.
And we have a question about, is the goal of treating MHS as a form of, sorry, just trying to word this, as a form of autism as primary treatment or adequate care? So I guess, should caregivers be focusing on MHS, on the MEF2C side of it, or kind of the supportive care side?
I
think
have
is
to
what
admit,
this-
I'm not sure I understand the question, but I think fundamentally, when I think of what we're trying to do, we're trying to help people, be their best selves. So in other words, I think most of the effects that we're seeing are through the brain and learning and behavior and thinking fundamentally. And so I don't think it has to be autism per se. I think it can be the behavioral manifestations in whatever dimension they take in that person. In certain people, there may be aspects of autism or autism-like behaviors that come forward in terms of expressive language and being able to connect to other people, in terms of whether there are certain rigidity or need for sameness because of just how they react to things. So if you wanted to call that an autism manifestation, yes, but I think there are other things that aren't specifically autism that we're trying to do to help support people as well. So I don't think of it as being, just autism or needing to be specifically autism, if that's the question.
Right, and that seems related to this question. Is it possible there could be treatments for downstream symptoms, for example, hypotonia caused by MEF2C, or is this project looking specifically for treatment at the gene source?
I guess fundamentally, I think of hypotonia as a direct manifestation of the brain, how MEF2C is affecting the brain in a very global way. And I would hope that many of the treatments that we would have, I don't know that we would target and say the outcome measure is hypotonia specifically. It might be that the way a child moves, how they move, how effectively they move as they're, controlling their body might be one of the things that we'd measure. And it might be through hypotonia or through improved muscle tone that we see that they're moving better, moving their body better. And that could be through, improvements in hypotonia.
And we have a few questions about funding in terms of families receiving funding. Is this an inclusive study, like removing a family's means as a barrier to participation, or are parents expected to pay their own way?
I'm going to turn this over to the foundation representation.
Hi. If we have any families that require funding to attend this research assessment procedure in Boston, they should contact us, and we would like to help fund any families that have difficulties to travel to this destination.
And we also have some questions about overall research funding. What do you see as the next need in terms of fundraising support? And how much money do we feel like we need to raise to complete the study for the cohort? Which is one of the questions.
So I'm going to say, in terms of the foundation, so you guys as foundation leaders, you should talk amongst yourselves about all of the many things you do. I'm sure this is only a portion in terms of doing that. We certainly are donating many things to be able to support this. For instance, I donate my time to be able to do this. The foundation doesn't get charged for me in terms of working with you, and I spend a lot of time with Anne and others trying to do these things. So there are a lot of things effectively you don't have to worry about. We do have people that are as part of the assessments for these. And so if the foundation covers travel costs or housing costs, hotel bills or something like that, you guys will have to figure out what that is. In terms of the assessments we do, though, there is for the evaluators that do this, we use some cores within Boston Children's Hospital. So people that just literally, it costs so much to be able to do a behavioral assessment. It costs so much for our physical therapist to do an assessment. And so that comes, round numbers, let's call it, if it ends up being something like a couple $1,000s per person who comes through for each time point they come through, as a round number. You guys can start thinking through as you're doing this. And again, it's ideal to do this with the same person on an annual basis to get some sense of what that trajectory, what that line looks like for their slope. So as we're doing that, I'm sure this will grow into something further. As there are opportunities with funding in terms of clinical trials, depending on who runs those trials, the hope would be that, there might be funding depending on the source. So if it's a company that's doing the trial, hopefully the company is actually sponsoring the trial. But, we'll see how this goes depending on who's developing those treatments.
And we have a question about whether all MEF2C variants would be eligible for a potential clinical trial, or is it possible that clinical trials would only be for certain types of variants?
So it all depends on what that clinical trial is doing. So I can't say for sure yet. I do think that in terms of the clinical trial readiness, we don't even know for some of the genetic variants how they're acting. So I think it's best not to leave anyone behind at this point and make sure that we're covering all of our bases. If the trial were something that we're looking at one subset or another, I think the other parts of the community are great comparators as we're doing this. So I think there are ways that the entirety of the information is going to be useful, even if it's a portion of people. And I'll give you an example that many times who's eligible for a clinical trial is not even based on the genetic variant, but it may be based on age. That is that oftentimes when we start things, the FDA requires that we start with older people, just knowing that something is safe. And then as it's proven to be safe in older people, they come on, in terms of younger people. And so, they might start out with something that's in adults, 18 and older. And after that, they might move it down to adolescents. And after that, they might move it down to children. And after that, they might move it down to infants, depending on what the safety profile looks like. So for multiple reasons, I think we want to be ready for any possibility of what either regulatory agencies might require, what the scientist is or the company is that might do this. I think it would be foolish to get too smart in these early stages and try and guess about what the opportunity is going to be. My hope is that there's going to be an opportunity for everyone. And like I said, I want to make sure we don't leave anyone behind.
And I think that's also related to this question. Will this meet criteria of FDA and EMA?
For baseline, yes. I mean, of course, we'll have to have the clinical trial and we'll have to know what the outcome measure is that we're going to have for that individual study. But the purpose is that this will serve as the baseline data or the comparator or the run-in data.
And we have a question about how is the research that we're doing different from others? And I think by others, they might be referring to the Simon Searchlight research. I'm not sure, but just kind of how is what we're doing different from other research going on?
So for Simon Searchlight, I hope I explained, and people may not have been here from the beginning, but that's an online measure. And so with all of that, that's really being reported by guardians. It's not direct assessment or direct observation. I probably didn't make this statement, but the idea is that those data come together. So the same person on Simon Searchlight for what they're reporting actually gets merged with information that comes from a direct in-person assessment for the natural history. So this Humpty Dumpty gets put together. The same person is known to be the different data sources, and that's how we know that we can validate a parent-reported piece of information.
And for in-person, how many visits will they need to make to clinic?
So my hope personally would be that this is an annual, so about every 12 months. It doesn't have to be precisely, absolutely on the day, but an approximately annual evaluation.
And how do parents best share information to the MEF2C scientific community that we are each individually gathering from researchers?
Can you say that one more time, Alexa?
Yeah, how can parents best share information back to the MEF2C scientific community that we are each individually gathering from researchers? So think families that have spoken to researchers, how can they kind of share back with us things that they have learned?
I guess I'm going to say that number one, as a parent organization, I hope you guys have mechanisms to do that. I think that's primarily what you want to do. And then for your leadership team, you guys as a leadership team frequently communicate with me. So feel free, any of you all, in your next email, let me know anything you want me to know.
We have a question about, is there a site where we can see what medications have been tried and how it has helped or not?
So I'll tell you what I'm used to doing, and if this works for you, I'm happy to do it, is that as families either in person or online, or a combination of the two, get together for having sharing meetings. We take the data that we have and present it back to the community. That would include medications that are being used. If there's a medication that seems to work or not work, we put all that data out transparently. We talk about what the life course looks like, so how things might be different as children are getting older. I will say that in general, I personally haven't seen a medication that's just like a wonder drug in terms of things. If any of you have, you should certainly share it amongst yourselves. But it gets to, I think, greater specificity of medication for treating what symptom and trying to get, more specific about that. And there might be medications for, gastrointestinal symptoms that, we do have things that are working for constipation as an example, but we may not in terms of issues that are related to behavior or learning or thinking.
And we have a question that this sounds kind of pretty exploratory at this stage. And what is the confidence that we will be more honed and ready for clinical trials in 3 years?
So certainly that's an accurate statement. And within the life course of MEF2C, I would describe this as still early days for the community. And you have to explore when you're in early stages of understanding a condition, understanding how it changes over time. That's just by definition how things work. There are absolutely no guarantees that in 3 years there's going to be something that's ready for a clinical trial. I want to be clear about that. I don't have a crystal ball. I myself am not working on a specific medication from F2C, but I do know many others who are, and they're good scientists and they're working hard at this. I'm just giving you the benefit of what I see working with many other conditions and seeing what's happened with them. And in some cases, my best case scenario is what we did with spinal muscular atrophy and how we really made incredible progress in a very quick time frame by being extremely intentional. And it was very much calculated in terms of how to make that work. And that absolutely, I'm convinced, saved the lives of a generation of children. So I don't mean to be overly dramatic, but if one is thoughtful and strategic about this, it can make a big difference. And I don't want your group to be left behind, because at some point there could be breakthroughs in technologies, and it could be many, many conditions that will run forward, or at least for whom these treatments could be available. And if you're not ready, there could be others that'll go ahead of you because they are ready. And so I just want to make sure that everyone gets a chance to be able to be ready. But I don't know if it's going to be 3 years. It might be 5 years. It might be 13 years. I want to be clear. I don't have a crystal ball in terms of knowing this.
And will there be an age cutoff for the study? This person's daughter is 21. In the next few years, will she be excluded due to age?
Short answer, no.
Is there somewhere that we can point our child's genetic doctor and counselors for more information on the current status and development of clinical trials?
I think today was meant to be that, so I don't have anything else specific.
And we'll have the recording of this today if anyone wants to potentially send that to anyone. Are there more children with MEF2C that might have been diagnosed incorrectly? This individual's daughter was not diagnosed until more advanced testing was done.
I certainly think the people that know their diagnosis are the tip of the iceberg of who's out there. So I don't know if they're undiagnosed or incorrectly diagnosed, but certainly I'm sure based on my experience with other conditions, I would be surprised if we know of even 1% of the MEF2C folks who are out there currently. And Alexa, we're just drawing towards the end of the hour.
Yeah, some of these we might have to potentially answer over email, might be possible. Let's see if I can maybe just pull one or two more questions. If our child's had a recent EEG, could we skip that part if we did an in-person evaluation in Boston?
The EEG we're doing is not a clinical EEG. So for those of you where your child has had a clinical EEG for seizures, those are technically different. We're doing something that tells us the power spectra and is in a very standardized way. So the short answer is no, we wouldn't have you skip that EEG in Boston. You would still do that. You don't have to do that glue, that smelly glue, like to your head. It's a cap that goes on relatively quickly and easily. So most people have done okay with the EEG aspect. It hasn't been nearly as hard as when you have it at your home hospital.
And I think this might be our last question in terms of time, but I know this is a question a lot of people have had about criteria to participate, that some parents who have a VUS have not been included so far. So kind of what has our criteria been?
Yeah, the VUS, one of the things that can keep you at a VUS stage is if the parents haven't been tested. Through Simon's Searchlight, we offer free genetic testing to the parents to disambiguate some of those variants of uncertain significance. And so I urge you to take advantage of that. Again, it's free to you to be able to do that. And so if you come through Simon's Searchlight as your first stage, go from a variant of uncertain significance to something that's clearer, then it helps us in terms of correctly categorizing you and being able to make sure that you're in the right club. Someone asked the question, what if you have the wrong diagnosis? We want to make sure everyone gets the right diagnosis.
And so I think going forward, since it seems we're pretty much out of time, any further questions about the Ascend study, the in-person, can be directed to that Ascend study email address that I gave. And really any questions about funding and the foundation, I think, can be directed to the foundation.